We understand that these times remain challenging. CMI continues its mission to make it convenient for you to get premier outpatient care while deliberately making CMI the safe clinic for you and all those who are dear to you. Premier outpatient care is now available in the tip of your hands. With the same unhurried, meticulous attention, you can consult with CMI's broad range of specialists by phone, by video, or in person. For patients who prefer to see their doctor in the clinic, virtual patient screening for symptoms is mandatory prior confirmation of your appointment. We send online health declaration forms for you to fill out. Once your appointment is confirmed, you are all set to go to CMI. Since companions are not encouraged, patients are dropped off at the main entrance of Ori Central, where CMI is located. Kindly keep your mask on at all times. We want you to be safe in our clinic right from the doorstep. Here are the clinic protocols we have implemented for your protection. Upon entering, your temperature will be checked by our care specialist. As we prepare your suite, you will be seated at our lobby while observing social distancing with other patients. You may get your snacks at our refreshments area, one patient at a time. Disposable cups for hot beverages have replaced reusable cups for the time being. Hand sanitizers and rubbing alcohol are also placed in key areas of the clinic for your easy access. Our doctors, care specialists, and staff are all wearing DOH-compliant personal protective equipment to ensure providing you with premier patient experiences. Your consultation is done in the privacy of a clean and spacious consultation suite. We also ensure to follow new normal processes for your physical therapy sessions and diagnostic procedures. Aside from our dentist and full personal protective equipment, we also use aerosol suction machines and acrylic covers during your dental procedures. If your doctor prescribed medicines, you may veer away from crowded drugstores as we have our pharmacy in the clinic for your convenience. We want you to be safe while in our clinic, even as we stay safe ourselves. All CMI staff undergo regular COVID-19 surveillance using Roche antibody screening. Lastly, CMI was conceptualized and built with your utmost safety in mind. CMI is on negative pressure airflow, and our high-efficiency particulate absorbing HEPA filters are built in. Since day one, we have been using UVC lamps to sanitize the clinic in between patients and after clinic hours. Also, our housekeeping staff regularly cleans and disinfects the consult rooms after each patient's use. In CMI, we are committed to provide the premier care you require and the safety you expect. Visit us at CMI for your outpatient medical needs.
Good afternoon, everyone, and we hope you are well. Centre Medical International, or CMI, is a premier outpatient clinic located in the heart of BGC. With over 150 specialists and subspecialists on our roster, CMI continues to be here for your medical needs, such as specialist consultations, by phone, video, or in person, diagnostic procedures, and preventive treatments, such as vaccinations and checkups. No crowds, no queues, no hassle. CMI is the safe clinic for you and all those who are dear to you.
Good afternoon, everyone. My name is Joseph Romero, CMI President, and welcome to our webinar today entitled Testing, Treatment, and Vaccines. Is there light at the end of the tunnel? I'd like to also extend a very warm welcome to our friends from the Management Association of the Philippines, ECCP, and SCHAM, and of course, our longtime valued patients and friends. Before I introduce you to our moderator today, let me just go through the webinar reminders for our common reference. So we have started exactly at 4 with the CMI video, and as participants, we have turned off your video and muted your microphones. However, we highly encourage you to use the chat box, the Q&A chat box for your questions later on. Please feel free to type in there whatever you need clarification on, and also indicate your full name as well so that we can properly address you later on. Without further ado, ladies and gentlemen, I'd like to introduce you to our moderator today, Dr. Hazel Zuellig. For those of you who have either been her patient or heard her give her very clear and brilliant lectures, we are honored today that she has agreed to be our moderator for this this burning issue of the day on vaccines, COVID-19 vaccines. She's currently in Europe at the moment, but she's made time in her busy schedule to help us sift through whatever may be technical or what may be too medical or too scientific in the presentation. And also, she's also here to answer some of your questions as well. So without further ado, again, ladies and gentlemen, Dr. Hazel Zuellig.
Hello. Good afternoon, everyone. It's actually morning here where I am, and advance Merry Christmas to everybody. This is going to be our last webinar for the year, and we thought that when everyone gathers socially distanced with their families and friends, or when in your Zoom parties, people will be talking about the year that was, and central to that is COVID. And we wanted people to talk about the right things, to dispel all the myths and the misinformation about testing, about treatment, and about the vaccine of COVID. So we invited the one person, to me personally, whose opinion matters. So let me introduce to you our speaker for today. Our speaker is Dr. Benjamin Ko. He was CMI's first president. He is a practicing pediatrician and a member of the Pediatric Infectious Disease Society of the Philippines. He graduated from the University of Santo Tomas and underwent his residency training in pediatrics at the UST Hospital. He did his fellowship training in clinical and molecular pharmacology at the Boston University Medical Center in the US. More importantly for our topic today, Dr. Ko was the former director of the Center for Drug Regulation and Research of the FDA. This is the office that is in charge of making sure that the drugs and the vaccines in the market are safe and effective. He is a fellow of the Philippine Pediatric Society, the Philippine Society of Experimental and Clinical Pharmacology, and also a member of the American Society of Clinical Pharmacology and Therapeutics. He is currently a faculty member for the course in Regulatory Science and Management at the Ateneo de Manila Professional Schools. Dr. Ko is an author of 46 local and international publications and edited numerous books in pediatric, pharmacology, and pharmaceutics. So we are really honored and grateful that he is giving us this time before Christmas to enlighten everyone about about COVID vaccine testing and how to treat it. Ladies and gentlemen, Dr. Benji Ko.
Hello, good afternoon. Merry Christmas in that part of the world, Hazel. And Thank you for the very kind introduction. I'm honored ikaw pang nag-introduce sa akin ngayon.
Of course.
All right. So without further ado, let me try to see if I can here. I will have to allow a moment. Okay.
I think Dr. Koy is having some technical issues. Um, we're trying to get him back. Everyone, we would like to welcome all the participants. For now, we have 90 attendees, so this seems to be really a hot topic. We received some advanced questions before we even started. We would like to thank those coming from the ADB and from all the other friends of CMI who have sent their advanced questions, difficult ones about the vaccine. And okay, I guess now Dr. Koy is ready to start.
Let me see if I can share my screen. Yeah, I got this. Sorry, I had to use a new laptop because my other laptop died. I'm sure you can see. Can you see the slides now, guys?
Yes,
Yes,
doctor.
Yes.
yes.
Right. Okay. So let's start with the talk. And I apologize for the technical glitch, but I'll try to keep this interesting and short. And I revised the slides a little from the last talk that I gave at the university. So this is a takeoff from it, and I revised some of the slides. It's really light at the end of the tunnel, and that's, I think, the burning question that we have, because we're going through a holiday season where pandemic fatigue, it it's actually real, but it has we feel it more as the Christmas holidays approach, and therefore we we now meet pandemic anger. Whatever we do with other people, they're really very hesitant and in denial that there is a pandemic around, or if they do accept that there is one, then they leave it to chance. And after 9 months into lockdown, it has actually come full circle. And pandemic fatigue is real because it's a state of being worn out by recommended precautions, restrictions that relate to a pandemic, and thereby it leads one to abandoning the precautions and risk catching the disease. The social norms are affected, mental health is affected, the economy is affected, and of course, pandemic anger becomes a consequence. And you can see that now because if you were here, Hazel, you would notice that the traffic is horrible. It's actually bad, and there are areas wherein the people would not even care less that the virus is still around. So we're trying to find light at the end of the tunnel. Hopefully, at the end of the lecture, I will shed a little more information if there is really, if we are near that light, because there actually is a tunnel that we're going through, and hopefully at the end of the talk, we will see that there is light at the end. And there is a path that makes us go from point A to point Z. And the path is actually that of testing, that of treatment, that of immunization, how to use rapid tests, and then there are new ones that are coming out in the market, how to utilize the data properly, and of course, combining all of these all together. So let's start with the first one, and the first one is testing. We don't test left and right, no? We need to do the right test. And the trace and track system, in order to reduce the effective reproductive number by 26%, is we find 80% of the cases within 24 hours. Unfortunately, that is not true, especially in the local setting, where in more cases than one, in many of the institutions, some of the tests come out 48, some as long as even 5 days later. But it is mandatory that we find the cases within 24 hours, and there is immediate testing following the onset of symptoms, and contacts are notified of their need to isolate within 24 hours. That trace and track system can only function effectively when at least 80% of close contacts are traced and told to isolate. The longer it takes us to find the people that we have come in contact with, if we are affected with coronavirus infection, then the more difficult it is to make sure that the pandemic is held at bay. So not enough contacts reached in time to make a difference in the effective reproductive number, and compliance with isolation guidelines is actually very low, particularly in the country. And that's the reason why I will show you in a little while, the curve has never really gotten better, unlike other countries where you see there's an increase in the total number, and then there is a flattening, and then another increase when another surge comes. Ours has really never declined. We are still in the first wave. And this is the Philippine testing data. You will see that over a period of time, on your left side of the screen, the curve actually is in multiple colors. When the color is in red, it shows you that we were not testing enough. That means that more than 10 to 20% of the cases were positive. On the other hand, as it becomes orange or it turns blue, then that means that there are less daily positive rates. And over a period of time, you will see that in the months of June, July, August, September, we were really doing bad, horribly bad. And we were reaching numbers up to 14, 15%. Or that means that out of every 100 patients being tested, 15 were actually positive. Now, if you see our curve, it's actually, in spite of the fact that we are testing less, we have less daily positive rate. Now let's talk about treatment. Of course, for any other disease, if you had to ask me between vaccine and whether a cure, which of the two is more important, I would always go with the cure. That's because with a vaccine, we are effectively able to prevent an infection, but it is not 100%. I'm sure it's the same thing with the cure, but to me, the cure is more important because any treatment to any disease is always the forefront in therapeutics. So we know that there was a WHO solidarity trial that the Philippines participated in, and they are actually not new drugs. They were actually repurposed antiviral drugs for COVID-19, and this is the result of the interim WHO solidarity trial, which came out early this month. And the trial showed that of 405 hospitals in 30 different countries, more than 10,000 adults underwent randomization to receive either remdesivir, hydroxychloroquine, lopinavir without interferon, and interferon. And there were 651 that received interferon plus lopinavir. And then, of course, there were 4,000 that were not randomized to any trial drug at all. And the results were disappointing in the sense that, well, this is just an interim analysis, but this is with over already 10,000 patients being involved. That remdesivir, hydroxychloroquine, lopinavir, which is an antiretroviral drug used for the treatment of HIV, and interferon regimens had actually little or no effect on hospitalized patients with COVID-19, as indicated by their overall mortality, by the initiation of ventilation, by the duration of hospital stay. So parang there was really not much effect for all of these. And what we need to do is we need to approach the treatment to COVID-19 in a different way. And there are three approaches actually to treating COVID-19. One is looking at drugs that keep the immune system from overreacting in ways that cause damage to the body. Two is using antibodies that can target the virus. Either these antibodies are synthetically made or they can be harvested from the survivor's blood plasma or made in a laboratory. Or we can use antiviral drugs that actually inhibit the coronavirus ability to replicate and therefore keeping the viral load low and undetectable. But of all of the treatment regimens for antimicrobial diseases, one of the most difficult is to make one against a virus. So let's look at the lessons from the United Kingdom. In the United Kingdom, did a trial on their own. It is called the Recovery Trial. And they used dexamethasone in the treatment of patients who are on ventilators or those who were on oxygen support. And they showed that in that particular trial, they can reduce the risk of death by 1/3 for those on ventilators and 1/5 for those who were on oxygen. Now, there were also antibody cocktails. There were 3 types of antibody-based regimens. One was a single monoclonal. The other was a cocktail of monoclonals. And of course, of polyclonal antibodies that were utilized as well. And while they work, they are far too expensive. So these antibodies are actually is what is in the pipeline. And there are a variety of them from, these are the ones that are in the pipeline. Regeneron was the one used by Donald Trump when he developed COVID-19 infection. Regeneron actually is an antibody cocktail, and it is made up of one synthetic antibody and one that was harvested from a patient in Singapore. And it has been assessed in a multi-phase clinical trial and has shown very promising results. This is the most promising of them all so far. Takeda has a hyperimmune globulin similar to other immunoglobulins, but this one is called hyperimmune globulin because it is specific for coronavirus. So it is called COVID-19. The drug is currently in phase 2 trials, but Takeda's approach is different from other companies because it relies on plasma that is collected from patients who have recovered from COVID-19. And therefore, it makes the manufacturing a potential bottleneck. But these are various antibody regimens that are in the pipeline. The bad news is for low capacity settings in low and middle income countries, and I talk about the Philippines, some of the therapeutics may be a more feasible part of a pandemic management strategy. And antibody-based are more difficult because of the cost, because of insufficient manufacturing capacity, and the challenge of the regulatory approval process among these countries. By the way, Regeneron is $1,200 a pop. So we go now to after treatment. Our best option is actually immunization because vaccinations and many forms of vaccines which work at preventing an infection reduce the severity of the infection at the very least. So that vaccination shift represents now the downgrading of severity and infectiousness of the virus if somebody who contracts that particular infection has been vaccinated. So when we get exposed to it, to coronavirus infections, then its severity and infectiousness decrease. And it is unlikely that it is 100% effective, because I've never seen a vaccine that is 100% effective. When we give measles vaccine, we give polio vaccine, herd immunity actually takes a big role in the immunization process. Because if you only vaccinate a few, but we don't vaccinate a sufficient number of the population, then we probably will not see the vaccines work to their full capacity. So what does the vaccine do? Number one, it reduces the risk of infection. Remember, the word I use is reduction of risk. It does not mean you will not get it. It significantly reduces the likelihood of developing complications, and because of that vaccination shift wherein the severity can even be made milder, then there will be less complications. If, for example, if you did not receive the vaccine, then and you got the disease, then that means that you would probably get more severe complications, or the disease would have been more severe in you rather than when you did receive the vaccine. The caveat here is it does not prevent one from becoming infected. And when one does get infected, one must still isolate, because one still is infective. Okay, so some people think that if they get immunized but they get milder infections, they are not infectious anymore. That's not true. Okay, so that means that you are still infectious if you do come down with the disease. So vaccines are not silver bullets. So that's the reason why I said therapeutics is still, to me, the golden rule here. And there is a need to find treatment for SARS-CoV-2 infection, except that it is more difficult to find the treatment than to develop the vaccine. Vaccines are not silver bullets to address the pandemic. They are part of the larger toolkit to manage and contain it. That means it is an add-on to other measures. And manufacturers entail pre-orders. I've worked with a pharmaceutical company in my past life, before I joined the government, for a short while. And it's the same story that we have. If before you can purchase even the local company is able to purchase any vaccine or any drug for the following year, you must make assumptions and you must do a pre-order. And those pre-ordered vaccines are charged to the local company, and therefore they should be prepaid. Pay first, immunize later. With regards to safety issues, I don't think that there are major safety issues in the vaccine for the short haul. All of the clinical trials that are being conducted anyway in the country and in other parts of the world now follow good clinical practice. We have learned from the past experiences of tragedies from clinical trials, and we now make sure that all of the clinical trials have ethical approval before they are conducted. So the approval process ensures that they are safe for use. The larger question now revolves not only on the safety, but more on the efficacy or the perceived efficacy of the vaccine. So we know that the vaccines are relatively safe. Most of the side effects are mild, particularly arm pain, fever, a few chills. Some of the more severe side effects are not that much compared to those, and they have no difference compared to those patients that receive placebo. So that means on the placebo arm, whether you do receive the vaccine or not, on the placebo arm, you don't receive the vaccine, the side effects or the occurrence of, let's say, Bell's palsy or the occurrence of a neurological complication is similar as when you receive the vaccine or the vaccine arm. So the vaccines are effective and the degree of efficacy will actually vary. You will need to remember that the immediate role of a vaccine is to protect our health and social care system and to protect those most at risk from severe infection or death from COVID-19. We've had 1.7 million people die already in the last 1 year alone. We celebrate the anniversary this month. So 1.7 million lives is not something that we sweep under the rug. And this is the current state of vaccines. And this is the latest update as of December 13. There are 40 in phase 1, 17 in phase 2, 15 in phase 3, 5 limited access. And most of the ones that are limited access are actually China and Russian vaccines. There are 2 approved, the Moderna and the Pfizer vaccine. One has been abandoned. We're looking at AstraZeneca also, probably seeking approval and most likely will get approved. The only thing with the AstraZeneca vaccine is the dosing regimen. The one that was abandoned was the one developed from Australia's University of Queensland. It was abandoned because most of the patients that were immunized later on were turning up positive for HIV, even though they are not truly positive for human immunodeficiency virus. So there is something in the vaccine that clicks on and puts them on a positive status, false positive, which means that it is not due to HIV itself, but secondary to the vaccine reading it as an HIV positive individual. Sanofi and Glaxo announced disappointing trial results. The combined effort of Sanofi and Glaxo showed that the vaccine works, is good, but in younger patients. And the target actually of many of these vaccines is in the older population, in the most vulnerable. So these are the leading vaccine candidates so far, and I'll go through them very quickly in a while. So the ones that are in the most excited phase, and everybody wants a piece of them, are the genetic vaccines. They're called genetic because it depends on the platform of these vaccines. So all of these vaccines have different platforms. You will see among the leading vaccines, the platform for Pfizer, BioNTech, and Moderna are messenger RNA, which means it is a genetic type. CanSino, Gamaleya, Johnson & Johnson, Oxford, AstraZeneca have a platform utilizing the carrier protein adenovirus. And then you have the protein vaccines, Novavax and Vector Institute. And of course, the inactivate, this is the old tested, this is the way most of the vaccines are developed, the inactivated viral vaccine. So you kill it, make it less infectious. In short, we inactivate it. And that is where Sinovac comes in as well. So let's look at the genetic vaccines first. The genetic vaccines are vaccines that deliver one or more of the coronavirus's own genes into our cells so that it provokes an immunologic response. And Pfizer's BioNTech and Moderna's vaccine are based on that particular platform. The difference actually is in the dosing. Moderna is 4 weeks apart, while Pfizer is 21 days apart, given intramuscularly. And the other difference lies in the storage, okay? And I'll talk about the storage between the two. Of course, Moderna has the advantage with 30 days refrigeration and 6 months at -20. The challenge for Pfizer's BioNTech in countries that have very poor cold chain facilities is real. And I'll discuss that very quickly with you. But let me give you an overview of what a genetic vaccine is. So how does the Pfizer vaccine work? And a lot of people don't know. They just know that there's a vaccine, but they don't know how that vaccine works. So let's take a piece of the coronavirus. And this is what a corona, that's why it's called corona, no? Parang there's a crown, okay? That crown, the SARS-CoV-2 virus, is studied actually with proteins that it uses to enter human cells. So para siyang wheel, no? And these spike proteins are actually the ones we target. They are tempting targets for vaccines and treatment. So once you develop something, you need to destroy these spike proteins. How do you destroy them? It's not just like shooting them like an arcade, no? That's not the way the world and the body works. But this is the target for the messenger RNA Pfizer vaccine. So that messenger RNA, all of us have that in our body. That molecule is very fragile. And our natural enzymes can destroy it immediately if you inject it into our body. So it needs to be carried in a way wherein it is protected. So the technology of Pfizer and BioNTech is to wrap that particular mRNA into what they call oily bubbles. And it's what we see as a lipid nanoparticle. Because of their fragility, that mRNA will always fall apart at room temperature. So they needed to make it stable. And that's the reason for freezing it all the way down to -70. Now, let's look at what happens to that particular oily messenger RNA. It enters the cell, and you have now a vaccinated cell. After injection, that vaccine particle bumps into the cell, it fuses into them, and it releases now this mRNA, translates it, and makes a spike protein. 3 of these spike proteins makes a spike, and these spikes and protein fragments now protrude from the cell. So yung original cell, wala yan. Okay, so after injecting, it will now mimic as if to each other infection. So some of the spike proteins now form spikes that eventually migrate to the surface of the cell, lalabas yan, and it will stick out of their tips. And then it destroys, of course, the vaccinated cell eventually dies. Pag namatay yung vaccinated cell, pag namatay, lahat itong mga nakakabit dito, lalabas na lang siya. Okay? It will float freely and they become debris from a dead cell. But the body has its ability to spot that particular intruder. And that debris will contain many spike proteins and protein fragments floating, which is now taken up by a type of immune cell. And that cell is called an antigen-presenting cell. That antigen-presenting cell engulfs the spike, digests the proteins, and presents that spike protein fragment. The helper T cells in our body attach to it. And together with a B cell, other immune cells, we have the T helper cells, we have the B cells. That B cell now bumps, those immune cells, bump into the coronavirus spikes, yung mga spikes na yun, it bumps into it, and protein fragments on the surface of the vaccinated cell. So ito, yeah. So it now bumps, it attaches to it, okay? So it now starts attaching to it. And the antibodies latch onto the coronavirus spikes. By latching on it, it destroys and prevents infection by blocking the spikes from attaching to other cells. Kasi may antibodies na tayo. Yung antibodies na yon, pag naka-block sila doon sa spikes, yung mga spikes na yon, hindi na magkakaroon ng infection or disease ng tao. Unfortunately, it's not perfect nga, because some of these spikes may not even get attached by all of the antibodies, particularly in patients who do not produce enough antibodies, because that's the way the immune system works. So antigen presenting cells, the one I showed you a while ago, has another ability. It can activate another immune cell. It's called the killer T cell. And this killer T cell now seeks out, okay? Our body has that ability. Para tayong our body is like a sci-fi movie. It seeks out and it destroys now any coronavirus infected cell that display the spike protein fragments on their surface. So it now kills those infected cells. So it has those two abilities. It stops the virus and it kills the infected cells. Beautiful vaccine, di ba? The Pfizer-BioNTech vaccine therefore requires 2 injections. It's given 21 days apart. It primes the immune system well enough to fight off the coronavirus. But because the vaccine is so new, researchers don't know how long the protection can last. But I'll show you the short-term data in a little while. Hazel had sent me the data for both Moderna and the Pfizer-BioNTech. So if you look at the Pfizer-BioNTech vaccine, this is the data on the BioNTech and the placebo group. This is 7 days after the second dose, and these were on patients who had no SARS-CoV-2. They had to be negative for SARS-CoV-2 before they were enrolled into the clinical trials. And you will see that of all the participants, only 162 in the placebo group developed SARS-CoV-2, while only 8 in the BioNTech group went on to developing SARS-CoV-2 infection. 5 of the 8 were between 16 to 55 years old, and 3 were more than 55 years old. So you see how nice the vaccine is and how effective it is, because its vaccine efficacy is anywhere between 93 to 95%. And over a period of time, the red ones are the placebo groups. So if you do a time analysis after 105 days, the protective efficacy of the vaccine, which is the blue one, is still present, while the patients that received placebo went on to just developing the infection eventually. So this is the cumulative incidence of the COVID-19 occurrence. In the red one are patients that receive placebo. The blue one are patients that receive the vaccine. This is after dose 1. And this is after dose 1, all available efficacy population. Let's look at the Moderna vaccine. This is more recent. The data came out December 17. Okay, so this is an interim analysis for the primary efficacy endpoint. Again, if you're looking at the vaccine group and the placebo group, while the Pfizer vaccine had 18,000, this one had 13,000 in each group. So this is about 26,000 or 27,000 patients. And if you look at all of the participants in the vaccine group, none in the 65 years old and above went on to developing COVID. And only 5 between 18 to less than 65 years old went on to developing COVID, while the placebo group mas mataas. Okay? So it has a vaccine efficacy of at least 94.5%. And you see the same trend, the occurrence after randomization, first COVID occurrence after they were randomized. This is an intention to treat analysis set. And you will see the blue are the patients that receive placebo versus the red are the patients that receive the vaccine. That over almost more than 3 months after the clinical trial was conducted, the cumulative incidence rate in the placebo group was much higher significantly compared to those that received the vaccine. Challenge for Pfizer vaccine in the Philippines is that it requires minus 70 degrees storage. One, two, it requires dry ice after each repacking. So some people were making the comment na we can replace the dry ice after every 10 days. Dry ice lasts only 24 hours. At best, if you have the right storage, dry ice will last 3 to 4 days. I mean, you know, the 10 days will not cut it. Baka naman wala na silang matanggap. So after each repacking, once you remove the dry ice, you will have to replace it with a new dry ice. And the box can be opened only twice a day. That twice a day, you can open it only for 60 to 90 seconds each time. And then when you need to immunize, you need, kasi frozen siya eh. Frozen, like ice cream, di ba? When we go to Magnolia, once upon a time, when we were little kids, yung mga iba na nakikinig dito na medyo millennial, hindi nyo naabutan yun, na we needed to buy ice cream together with dry ice. So frozen yan. But you need therefore to thaw it at room temperature for more than 30 minutes. Once it's thawed, you should administer all the vaccine within 6 hours. So it becomes a logistical nightmare, if not challenge, to utilize this vaccine if you don't have all of these things that I mentioned taken into consideration. Diba? Kasi sometimes you have patients that volunteer and then later on, they, laban bawi yan eh, laban, bawi, laban. Oh no, second thought, no, I don't want to go. Oh, do I go? Don't I go? Do I go? The next type are the viral vector vaccines. The viral vector platforms actually are the CanSino and of course Sputnik 5. You'll see it because of their names. You'll see their AD, which means adenovirus. They contain viruses that are engineered to carry the coronavirus gene. So they are called carrier protein molecules, and some of the viral vector vaccines enter the cells, cause them to make the viral proteins, and other viral vectors slowly replicate, carrying coronavirus proteins on their surface. The nice thing about it is they're okay in the refrigerator or just freezer storage. AstraZeneca Oxford vaccine is an adenovirus vaccine. It's called AD10X. It's a chimpanzee adenovirus vaccine. So its efficacy is up to 90%. The best case scenario is give half the dose now and then the full dose 4 weeks later. So they have two parts of the clinical study. In phase 2, they were looking at full dose, or half dose, full dose. And voila! They found out that the half dose, full dose had better efficacy compared to the full dose. So I think they will end up with recommending half the dose and then the full dose later on. The beauty of the Johnson & Johnson vaccine is it just needs a single dose. And they are, again, ideal vaccines because they are stable in the ref. For protein-based vaccines, they are vaccines that contain coronavirus proteins, but there is no genetic material in them. Some of the vaccines contain whole proteins, some contain fragments of them, some pack many of these molecules on nanoparticles. And Novavax and Medicago are those vaccines. No? They're given 3 weeks apart, the same as the Pfizer BioNTech vaccine, and stable in the ref. And of course, the clinical trial, which is currently being conducted in the Philippines that has been approved by the Single Joint Review Ethics Board, is the Clover Biopharmaceutical vaccine. They've just finished their phase 1 and are now in their phase 1 and 2, combined phase 1 and 2 clinical trial. They have worked together with GSK and Dynavax. GSK is the British drug maker and Dynavax is an American company. The reason for their collaboration is that the diluent that they are using for the vaccine is ASO3, aluminum sulfide. And aluminum sulfide is beautiful in the sense that it's an adjuvant and a diluent at the same time, the diluent for this boosts the immune system. So it stimulates the immunological response to the vaccine. And therefore, it's a collaboration between Clover, GSK, and Dynavac. And of course, lastly is the inactivated or the attenuated coronavirus vaccines. These are from weakened coronaviruses and coronaviruses that have been killed with chemicals. This is the conventional way of making the old vaccines in the past. The inactivated polio, the inactivated Hib, and all of the other inactivated vaccines that we give for children for their childhood immunizations. The two vaccines, one by Sinopharm and one by Sinovac, are approved already. This Sinopharm is used in the United Arab Emirates. I don't know if that's one of the reasons why they're very, very low cases already. And Sinovac has its limited use also in China. They give it to healthcare workers and the frontliners and their military. And I don't know if that's also one of the reasons why they have almost only about 30 to 40 cases a day. Okay, so we're done with the vaccines. Let's look at the role of rapid tests. I know I'm talking too long, but I will try to wrap it up in the next 20 minutes. So the role of rapid tests is that the key to upscaling testing capacity plus addressing test and trace challenges is bringing at least on stream all the possible rapid antigen tests. And there are more antigen tests and there are more rapid tests that are in the pipeline. And the day I would like to see is that some of these PCR tests that we can get results in 90 minutes is being developed. You have the RT-PCR technology from NudgeBox produced by DNA Nudge, and you get results in 90 minutes instead of 5 days or 2 days or 24 hours or even within the next 12 hours. I mean, 1 hour and 30 minutes is makes a lot of difference. And then there's the LAMP testing. The loop-mediated isothermal amplification turns the plate readers into diagnostic tools. There is CRISPR, which is also in the pipeline. It is programmed to detect specific sequences of the DNA in the SARS-CoV-2 virus by snipping the gene of interest, and the results come out in less than 20 minutes. And of course, Curative, which is the company, not the curative, no, saliva testing is not curative, but the company is at the forefront of coronavirus detection and offers a saliva-based COVID-19 test. This is out of Israel, and this is beautiful for mass testing. We still have to see whether it is applicable in the local setting or not. The challenge to organization, what is clear on testing is the challenge to organization, not the supply, but the challenge of organization. The critical step is organizing how these tests are used. One is to ensure appropriate tests are made available. You know, we get rid of some of the tests that are not really useful and know which and when to use. Support and ensure the lab processing facilities. Like there are 76 testing centers in Metro Manila alone, in the National Capital Region. Of the 180 testing centers in the country, you can imagine that almost half of them are in NCR. And there are very few actually in the more challenging places outside, in provincial settings. That's why in some of the regions, it takes them quite some time to get the tests back because they will have to send it out to another nearby region that has the test. But it's all about money because it's the private sector actually that does the tests. Because nobody is willing to shell out money to have the tests done on the government level. If there are some on the government level, they're not enough. And if you should put in place the right testing regime, those that need to isolate should be given the right support financially and access to further testing to ensure isolation only for as long as needed. You don't need to isolate them for the longest period of time, and you should give them the right incentive. They're out of work instead of just putting them and locking them up, because they will not want to isolate themselves if they will lose their jobs. Data from all those being tested is captured and stored centrally. So there must be simplicity in communication and clarity in the incentive. And it is important for the government to clearly communicate the purpose and the mechanics of the testing regime. You have to tell them what a negative test result affords and what a positive result means, not only to the individual, but to the community at large. Lastly, we talk about the data. No? When I talk about the data, and I love talking about data, it's actually the numbers. We have to always be truthful when it comes to data. And when we lack robust data platforms, then it creates chaos, because the local government unit and the national government unit do not agree on what the data is. The data has revealed many of the shortcomings of our systems, from being able to effectively monitor the disease to allocate and distribute tests, and later on, eventually, the vaccines. Data is central. Doon mo lang malalaman kung kanino dapat i-appropriate yung vaccines na to. Sino ba dapat mauna? Saan ba dapat natin ilagay muna? Saan dapat nakacentralize yung mga tests? In essence, the pandemic is ultimately an optimal control problem. A complex dynamic situation where decisions in one stage will impact not only on the outcomes, but also on the constraints for subsequent decisions. So this was yesterday's data. I'm sorry, I had no time. I was so busy today. Put today's data. Normally, I give you the most updated one. But if you want to know today, we are 77.7 million coronavirus cases already. And I used the one last December 11 to show you what one country's data could do to upend all types of data. So in one day, on December 11, we saw 1.5 million cases. It was a new world record, and it was not something we were looking at. If you look at the death rates, it's all the same. No? But if you look at the jump in the new cases, Turkey posted over 800,000 new cases in a single day after it started to report better numbers. Why? Because all the numbers it reported were only for symptomatic cases. They were not reporting patients that were positive but had no symptoms. So when they did that to report everybody that was positive, they released everybody. We had 1.5 million cases that day. And so this is the data curve of Turkey. You see, one day they shut up. So that's what happens when you don't tell the whole story. And you can also tell by the European Center for Disease Control, this is how the data of Turkey looked. All right. On one single day, and then eventually going back all the way down to what it normally should look like. And as of December 20, this is an overview of how the countries in the world are performing. We are in an upward swing. Many of us are in a second or a third wave. India is in its first wave and it is going down. You see? Japan is in its third wave. It went down, then went up. And because it went down, but it did not go down significantly, it is on an upward trend again, with Japan having almost more than 2,000 plus 2,500 cases daily in the past week. And this is what it looks like in selected Asian countries that are seeing a surge. South Korea is seeing its third wave. Indonesia is like us. It has not gotten away from its first wave. Japan has gone from one wave to another. The Philippines this is the Philippines. We went up. This is a fluke, the downward here. No, that's not real. And we're starting to go down, but we are on an upward trend. After the Christmas holidays, if this upward trend keeps going up, then we are in our second wave. Because it's very hard to bring down a second wave. Why do I say that? Because remember, the first wave started from baseline 0. If you get a second surge from baseline 1,500 cases, look at what happened from baseline 0 sa atin. We reached 6,000. What will a baseline of 1,500 mean? Okay, so I give that back to you. And this is our Philippine data. This is the slight uptick in the last 7 days. Today, we have 1,300 cases. That's a slight decline from yesterday's 1,700. But that's not nice, because if you drill it down, the National Capital Region has seen a dramatic increase of an average of 300 plus cases a day to almost 500 to 600 cases daily. So that's almost a double. And the Christmas surge has just begun. So you won't see the cases until after the New Year. This was our data yesterday. While we saw less cases, this was down from 1,750 plus. So there were just 30 plus less cases yesterday than the previous day. So what type of data do we need to collect? We can't be collecting everything in the world. We need to collect incidents, the graph, who has been near who, when, and what circumstances. We need to collect clinical data, how many are in the hospital, in the ICU, whether treatment is effective or not. We need to collect quarantine and isolation data. When does their time expire? When can they go home? Are they being checked or are they compliant even if they go home? And once we start immunizing them, there should be a central database who has been vaccinated. We need to collect the names of these people. You know, we have a lot of informal settlers. That's one of the reasons why we're not able to bring the cases down. It's because the top areas that are in the National Capital Region also have the top most number of informal settlers. And I hate saying it, but unless we answer the problem of the informal settlers, you will not be able to bring the cases down to 0. And 0 is the ideal number. Okay? So when we start immunizing them, hindi pwedeng yung isa mawawala kasi sayang yung bakuna. Okay? And the vaccines are not interchangeable, not at the moment. And I don't think they will be, because they are coming from, as I explained to you a while ago, different platforms. So we need high-resolution real-time data, which allows for precise and effective policy making. It ensures that decisions are not impacted by lags in reporting or based on forecasts. And we've tried forecasting. I tried that in the first few months, and it is unreliable because of the situation on the ground and how people individually react to good news or bad news. The ideal world of policymaking looks at lockdown levels, contact tracing prioritization, test allocation, vaccine allocation, risk calculus, and shielding. And we should remember that there should be reprieves in between with lockdown levels. Hindi ka pwede parating nasa ECQ, nasa GCQ, hindi kayo barbecue. No, all of these have to come to a realistic level. And the problem with the National Capital Region is that they are all interconnected cities. 17 cities are all interconnected with one another. If I want to lock down Quezon City for the sake of putting it into firmer action like locking down Wuhan, I would have to lock up 2.3 million people, including Congress and all the congressmen that live within that area, which makes it a little more difficult because they will not be able to travel in and out of that particular city until we have wiped out any coronavirus case in that particular city. But people live there or work there. See, that's the problem we get with the National Capital Region. And then contact tracing prioritization, 2 to 4 days of waiting for results does not cut it. It should be there within the next 24 hours. Otherwise, the longer we get the results, the more poor contact tracing is. Test allocation, we should allocate more in areas where there are less testing centers. And of course, we should try to avoid trade-offs with private corporations, because the government should be fully responsible for the allocation and distribution. It cannot be one for you, one for me. That I'm going to buy, but I will buy for my people. You can give, I will give the other half to you to distribute. Then, of course, we need to take into consideration the threat of COVID versus other illnesses, placing COVID-19 into perspective in terms of relative impact on public health by comparing it to other leading causes of death and disease. We must remember that other diseases are prevalent as well. And COVID alone has set back progress on eradicating other preventable infections by disrupting immunization programs in many countries. So simplicity is here. Simplicity in communication, clarity in incentives is the key, and it is king. And there should be no room for confusion. And confusion only leads to building more panic and more pandemic anger because of the pandemic fatigue. You're tired, we're tired. Where is everything going to? So moving forward, the 3 keys to the light is testing, therapeutics, and immunization. I've summarized that. And we have never been in a better situation for a pandemic, because we've seen what technology and the advances in science and molecular biology has done. That in the last couple of months, we've come up with a vaccine that works. The question is, how effective will this be, and how long can this be protective? Because you need to immunize a lot of people, because it has affected more than 200 plus countries worldwide. Of course, as if our problems are not enough, comes another news. There is the mutant SARS-CoV-2 out of the UK. And the UK coronavirus, what we do know here is what we know for today. Kasi ang dami magtatanong, sasabihin nila, paano yan yung bagong variant galing sa UK? Okay, it's called variant B117 or B.1.1.7. They were actually, the UK saw a rise actually in September, and that particular variant was collected from patients last September. There were relatively large number of mutations, 23 all in all, that were acquired. And we see it as a potential problem on how the virus spreads. The current epidemiological evidence shows that this variant is very good at spreading. So this is that new variant. You see, this is still the coronavirus on your right side. And the spikes, you see, the spikes are used to latch on to enter every human cell. And all of these spike proteins now have this variation here on the right side. So the mutations that led to the B.1.1.7 variant is an ORF1b protein. Okay, so at this point, that spike protein now, there is a change in the sequence of the ribonucleic acid. Instead of reading it as UAC, it is now read as CUA. So there is a variation in this end of the spike protein on the coronavirus genome. Okay, so there is also a change in the amino acid. You see, meron na siyang lysine. Yung original, wala siyang lysine. Puro isothreonine lang siya, tyrosine. So what is the implication of the new variant? One, there may be an increase in transmission, at least partly the result of how it infects children. Because normally children are less likely than teenagers or adults to get infected or pass on the virus. But the new strain can potentially make children as equally susceptible as adults. But let's put a little story back into the 614G. Remember, a few months ago, we reported also that there was a new variant. It was called 614G. It is more transmissible, but discipline containment measures such as the physical distancing, masking, washing of hands, and what we currently do in the apat dapat still contain the virus, and measures work just as well. So are we talking about a more contagious? We don't know. Okay, we don't know for now, this new variant. We know that it can probably replicate much faster, but we don't know if it's really more contagious, because if you look at the setting in the UK, the outbreak actually occurred in a central part of London, in the UK, wherein it made the virus more easily infective because of the density of the area. And when variations occur, sometimes they kill themselves. No, some of these viruses are suicidal. So a new variant may mean a weakening of the virus. They are more transmissible, but they are weaker, right? So we still don't know that. We hope that that's the case. So there's no strong evidence that it causes more severe disease. Preliminary studies show that people infected with this variant carry a heightened, mas mataas yung viral load at yung higher concentrations in the upper respiratory tract. And we know that more viruses can create more severe symptoms, but are we seeing that? We don't know. For the moment, we don't know if there are more severe symptoms. We're seeing more cases, but we're not seeing more deaths. And the question is, how did this come about? Well, the answer is no one knows. The one in bold here is the typical infection. So typically, we get infected with the virus, we become infectious for a few days, and then we show signs and symptoms. Later on, the virus becomes less abundant in the body as we mount an immunological response. And of course, that immunological response, which I showed you a while ago, clears the virus in a few weeks. With a weak immune system, some of the virus thrive for months, and they accumulate large number of mutations as it replicates for a long period of time. So sometimes some of the patients actually with weak immune systems or who have lived through the viral infection, sila yung dahilan kung bakit may mutation, because it's possible that they can still replicate and they can still transmit some of the viruses that have stayed in their body for quite some time. The other possibility is drugs, because we put some of the patients on experimental drugs. Could have they been responsible for the evolution of the variant? I've always said, and I've always told my students, that microorganisms have lived longer than us. We've killed them, they've resurrected. We kill them, they resurrect. We start antibiotics, but they've seemed to find a way, antivirals, they've seemed to find a way to thrive, to exist, to coexist with human beings. And that's when they develop resistance. They mutate on their own. Then, of course, there's also the spread through animal population. That I'm sure you've heard of the story of the minks in Denmark, the civets in China, and other animal reservoirs. And of course, the burning question will always be, and inunahan ko na kayo para hindi na kayo magtanong, will the variant render the new vaccines ineffective? Most likely not, and I don't think so. Now, it's conceivable that a mutation can change the shape of the spike proteins, which makes it harder. And that's why I wanted to illustrate to you a while ago what it looked like. So it can make it harder for the antibodies, but pag pinalitan mo yung shape, it will make it harder for the antibodies to gain a tighter grip on them. But our immune system has a range of antibodies, whether they are T, B, they are the killer cells. That range of antibodies against a single viral protein can make it less likely that viruses can easily escape their attack. Someday, somewhere, probably, the vaccines may not seem to work as good as it should. We really don't know, but we'll know sooner or later. For now, even with the adaptation of the variants, will not make the vaccine useless, because as I lectured a while ago, because of the fact that our immune system in itself will make sure that it is adaptable to the shape in the spike proteins. What can happen, though, is it can become less effective. But as to whether it is ineffective, I don't think so. For now, we don't know, but we will know soon because as science evolves, so does the virus. And therefore, continued vigilance is important. And until we know more, it is wise to continue to follow all precautions. Yung apat dapat, ba? Airspace is important. Better in open air than a crowd. Physical distancing is also vital. Always wear a mask. When appropriate, put a shield on. And the time that we are conversing, eating, and doing all other stuff without it should be minimized to 15 minutes at most. So with that, I hope I gave you a clearer picture on what there is. And I wish you all a blessed, safe, and Merry Christmas. Thank you.
Thank you, Benji. I learned so much. That was more than 1 hour of solid immunology, epidemiology, virology. Very, very clear. Thank you very much. I'm getting a lot of messages. Everybody is just so impressed. And I have so many questions for you. So we're going to do a rapid fire, Benji, because in the interest of time. One question. What is the difference between emergency authorization and full approval? When the FDA, like in the US, UK, Singapore, they gave emergency approval for Pfizer and Moderna. What is the difference between that and a full approval?
Well, an emergency approval means that they've not reviewed all of the data and that the companies should still continue to submit ongoing data, which is what the clinical trials actually do. If you look at all of the clinical trials that have been approved, the patients are being followed up even up to a year, which means that, all right, phase 3 ka, approved na yung kwan mo for emergency purposes. So in the exigency of the current pandemic, your drug is approved, your vaccine approved, but we will continue to monitor it until the last patient or until the end of your clinical trial. Yeah.
Okay. But that means the government is responsible for distributing and dispensing this vaccine.
Yeah, because it is on an emergency approval. If you ask me, on an emergency approval basis, then it should be the onus of the government to make sure that all of the vaccines that are at hand are monitored properly. That's the reason why I was saying it. Sometimes when you have both the private and the public sector sharing it, they must have one platform. I'm not against them sharing it. What I'm saying is they must have one common platform. And what's very important here also is that they must have the same common ground for utilization, which means that it should be it should be one that is ethical. I know you know what I mean, no? Sometimes the least and the last will also be the last to get the vaccine, but that is not fair.
I know. So that's why we want to make sure that there is an equitable vaccine distribution all over the country, such that the most vulnerable, the seniors, the healthcare workers, and the poorest, who have already very poor access in everything, will have first access in these vaccines. Okay, if we move to the other question, somebody asked, if you only are able to get the first dose and for some reason you don't get the second dose, what is the effect?
Well, then you get also partial immunity. I cannot guarantee you because all the studies end with a second dose. All right. So the interim analysis show that the first dose works, but you will notice also that they required a second dose to get the full effect. So, and the second dose was given actually at a very short interval. Normally, you'd give one now and then the next one 2 months later or 6 months later, di ba? This one, no. 3 weeks talaga to 4 weeks ang interval niya, which meant that they were not very confident in the effect of a sustained immunologic effect
With
in
just one dose.
single dose.
Yeah. Now, with this variant now coming out, can you see looking forward that the COVID vaccine will be like the flu vaccine, something that we will have to have every year now from now on?
Well, until all of the clinical trials are in, after we see the results, because the ones that I've reviewed for the clinical trials that are being conducted, though, you will notice that their clinical trials don't end yet. That's why it's emergency use access, and that they continue to monitor all the patients. If there is a sustained response, as you can very well see, maganda yung sustained response niya after the second dose, even after 120 days. Then we will know after a year if the immune response declines or that there are more cases of COVID in the patients receiving the vaccine arm. But for now, the good news is that we probably will not need a second dose yet. A booster dose yet. But we'll see how it goes because you know how mutants are. It's like the flu vaccine. Diba? We have to add and we have to change the strain annually.
Okay. Somebody was asking about the clinical trials that, ethnicity plays a role. So how many, in your review of the clinical trial data, what is the percentage of Asians among the population that was tested? Meaning, will FDA require a clinical trial just for Filipinos? I heard that FDA announced that as long as it's of the same ethnicity, they will approve the the vaccines in the Philippines.
Well, it's kind of silly kasi Singapore did not require ethnicity studies.
Yeah. That is right.
You know, you don't need to do that anymore because ako, kaya nga emergency use access eh. Why else do you want to do the ethnicity study? You have to remember that many of those that were included in the clinical trials for Pfizer and BioNTech were actually Filipinos. And I have a lot of friends that joined the clinical trials and they are Filipinos. They're not Americans. They're American citizens, but they're not Filipinos. And about 13 to 15%, I believe, were Asians.
Okay.
So
Okay.
that's kind of a lot.
Yeah, that answers that question. A lot of questions about vaccine distribution. I think the government there came up with their priority list of who should get the vaccine first. And of course, the non-seniors and the non-healthcare workers are wondering, and they were hoping that the private sector can get vaccines also for them. So what is your comment on this?
Yeah, the vaccine naman can be given to the private sector, but like you said, no, equity first. So unahin natin yung talagang nangangailangan. Those that really need it most.
Hindi yung may pera lang.
Yes, because you know,
Sorry,
we all want to get out of this one, but we have to do it. We have to do it charitably, equitably. The common denominator here is we start immunizing first the vulnerable population. After the vulnerable population have been immunized, there should have actually been a plan. And that plan is what is the next batch and what regimens do we provide all of these people? I think that's the most important thing. Like how many million do we get? Okay? Sino ang unang bibigyan? Tapos, at saka, hindi naman lahat may gusto eh. Kasi like in the UK and I think in Europe, it's the same thing, even in the US. Not a lot of people wanted to get immunized.
50%.
Yes, correct. Yeah, 50%. You know, the Philippines, in the Philippines, somebody And the SWS did a poll and said 2/3 of the Filipinos are willing to get. I did a poll, and 50% of the people that I did in the poll actually wanted to get the vaccine. The other half did not want. And my poll was more on doctors. So the doctors were willing. But the patients, sige, kayo na muna, doktor.
Yeah, somebody said to me, I don't want to be the first because I don't want to be the guinea pig. So my comment was, the clinical trials, that's where the guinea pigs were. And once it is approved, you're no longer a guinea pig. But I guess if you have 50% vaccine hesitancy, then 50% are willing to take it. And if we are able to immunize 50%, at least maybe we can get herd immunity.
Well, you need to immunize 50% to 7. Actually, you need to immunize about 60 to 70%. Yeah.
So we need to convince another 20% of the population to get the vaccine.
Yes, we need to convince them.
Now, talking about vaccine hesitancy, one of my last questions, somebody was asking about the side effects. You mentioned them. And then, of course, with everybody publicizing the allergic reaction here and the Bell's palsy here, somebody asked about these vaccine injuries. Who should be made responsible?
No one. Actually, because it's emergency use access. So that means they will always ask you if you want to get immunized. And it's an informed consent. And it's a written informed consent. Are you willing to get immunized? I'm not forcing you. And therefore, if you develop an adverse reaction secondary to this, because it is already approved, then no one is held liable for any adverse event, serious or non-serious, that occurs with you after.
Yeah, it's voluntary.
Yes.
And everything, like in all the medicines we doctors prescribe, it's always a risk versus benefits. I guess FDA approves the drugs because the benefit far outweighs the risks in this vaccine. Yeah. All right. I think with that, we can wrap this long webinar.
But the other thing is that some people always ask, kailan ba yung mga bata? I keep telling them, they're not included in the clinical trial. So it means I think there was to conduct a clinical trial for them eventually. Children. There's one country that's doing it, Indonesia, for Sinovac. They're doing the clinical trials for children.
For children. Yeah, because I think there was even this big split in the US about whether to cut it off at 16 versus 17 years old.
Yeah.
Okay. So this is something that we're all learning about. Maybe in a few months we will have another webinar on what is our vaccine experience. We don't know. In the meantime, as you said in your title, is there light at the end of the tunnel? You said yes, there is light at the end of the tunnel, but it is a tunnel that we have to go through first. It is painful before we get there. It's not magic. It's not going to go away now, but we will get there. I guess for everyone, that this new year be a time of some optimism. We lost a year. We lost a year of our lives, but maybe with all of these things that you discuss, we will see the new year in a new perspective. So Merry Christmas, Benji. Merry Christmas to everyone, and I miss you all in the Philippines, and be safe, be well, enjoy the holidays, but please remember to be safe. Thank you.
Thank you. Merry Christmas.
For Thursday to December 27, Sunday. We will be open on December 28, Monday to December 30, Wednesday from 8 a.m. to 4 p.m. We will be closed on December 31, Thursday to January 3, 2021, Sunday. Regular clinic hours will resume on January 4, 2021, Monday. Visit our website www.cmi.care or visit our Facebook page facebook.com/cmi.care for the latest updates.